The basic structural unit of all living organisms is the cell. In humans specifically, it is a eukaryotic cell. Let's take a look at one now. Components of the eukaryotic cell are involved in many essential functions in multicellular organisms such as ourselves. Inside the cell, there are subunits called organelles, which all perform different functions, such as the ribosomes and the endoplasmic reticulum shown here. The organelle that is the control center for the cell is called the nucleus. The nucleus contains deoxyribonucleic acid, or DNA, which is the blueprint of life. DNA contains all the genetic information and instructions for the development and everyday functioning of the organism. Every single cell in a eukaryotic organism has a copy of DNA, and that DNA is copied every time the cell replicates and divides. Let us zoom in on the nucleus so we may better understand the process of DNA replication. DNA is made up of two chains of molecules called nucleotides, arranged in a double helix pattern similar to the winding of a spiral staircase. The nucleotides are like stair steps between the two backbones of the helix. To better illustrate the structure, let's unwind the DNA to reveal the nucleotide pairs. There are four types of DNA nucleotides: adenine, thymine, cytosine, and guanine, usually referred to as A, T, C, and G. These nucleotides are known to selectively bond to one another, such that adenine, A, consistently bonds to thymine, T, and cytosine, C, to guanine, G. Adenine and thymine pair together with 2 hydrogen bonds as shown, and cytosine and guanine pair with 3 hydrogen bonds. This selective bonding is why DNA replication is referred to as semi-conservative replication. This means that each single strand of the DNA can be used to make a new double helix. In order to replicate the DNA, this double helix needs to be unwound. An enzyme called DNA helicase, which is an ADP-dependent enzyme, unwinds the 2 strands, separating them in a way that is similar to unzipping a zipper. After the 2 strands have been separated, each strand serves as a template for synthesizing a new complementary strand. This process is possible because of complementary base pair pairing, where A's pair with T's and C's pair with G's. The next step in replication involves DNA polymerase. The job of DNA polymerase is to add complementary nucleotides to the new strand in a 5' to 3' direction, resulting in 2 new double helices identical to the original. The terms 5' and 3' are used to give directionality to the strands of nucleic acid. The 3' end, or the third carbon from the oxygen, in a clockwise direction, will always have a hydroxyl group attached and is where the next nucleotide will be added to the strand. The 5' end, or the 5th carbon, clockwise direction from the oxygen, will always have a phosphate group attached. The central dogma of molecular biology states that genetic information flows from DNA to RNA to proteins. The process of going from DNA to RNA is called transcription. Normally, DNA is tightly condensed and gripped by histone proteins. This prevents any transcription factors from accessing the DNA. To make the DNA accessible, the histones undergo acetylation, causing them to loosen their grip on the DNA. Now that the DNA is no longer tightly wound, transcription factors can bind to the promoter region of the DNA. One difference between DNA and RNA is the ribonucleotide uracil. Instead of the thymine used in DNA, RNA strands use uracil. When making the RNA, uracil pairs with the adenines on the DNA, and cytosine and guanine still pair with each other. Similar to DNA polymerase, RNA polymerase is involved in the addition of ribonucleotides in a 5' to 3' direction. Once DNA is ready accessible by RNA polymerase, the polymerase attaches to the DNA strand on the promoter site. Promoter sites are special coded sequences on the DNA strand that tells the RNA polymerase to start transcription. Once the RNA polymerase has recognized the promoter site, elongation of the RNA strand starts to occur and continue until a special sequence initiates termination. After being transcribed, the new RNA strand undergoes post-translational processing to become messenger RNA. The modified mRNA then exits the nucleus and goes into the cytoplasm where the next step takes place. We can now move on to the next step of genetic information flow, from RNA to proteins. This process is called translation. Another organelle is needed for translation, the ribosome. The ribosome attaches to the mRNA and reads the mRNA 3 base pairs at a time. Each 3 base unit is called a codon. Each codon instructs the ribosome to add 1 amino acid to the growing polypeptide chain. These amino acids are carried by transfer RNA or tRNA. Each tRNA has an anticodon that is complementary to a codon, and each tRNA always carries the same amino acid. The ribosome can read the codon, and the tRNA with a complementary anticodon adds an amino acid. As each codon is read, more and more amino acids are added to the chain until the termination code is reached. Once the chain of amino acids, called a polypeptide chain, is released, translation is complete. To complete the last step and finally to become a protein, the polypeptide undergoes complex 3D folding that is essential to its function. Finally, the process from replication to translation is complete.